Thank you guys for attending another MCBC training session today. This is the first of 3 GeneLab molecular biology training sessions. Thea is the manager of GeneLab. She's the one that's basically helped organize all of these. Today she's arranged to have John Pfeifer, who's a senior field application specialist with Applied Biosystems, come in and talk to us about fundamentals of real-time PCR. And some of the Applied Biosystems platforms in performing real-time PCR. With that, I'll turn it over to John to get started.
Thank you, Mark.
Yep.
So, my name again, John Pfeifer. So I'm going to try to figure out if I can, there we go. So just to let you know, this is the first of a 6-part training series that we offer free on our website called the Real-Time Quantitative PCR Training Series. So this is part 1, and if you'd like to go through the other parts, certainly go to our website, and it's completely free. Moving on, the assumption here is that you're familiar with traditional or basic PCR principle. So the PCR is a method to amplify specific genetic sequences. And that in traditional PCR, we are detecting the product using endpoint methods, such as gel electrophoresis and blots. However, in real-time PCR, we detect the product automatically each cycle. To do that, we need both a real-time instrument which combines a thermal cycler with an optical detection system, as well as a real-time fluorescent chemistry. So the chemistry is such, it will emit a fluorescent signal in direct relationship to amplification. There are some clear benefits to real-time technology. First, we get a lot more we don't have to worry about the post-PCR procedures. They're traditional and gel-based PCR, so gels are gone. That's nice. We get a lot more information than gels. We get to see different data layers, there are different dyes in the chemistry, and we get to see all 40 cycles of data. But where real-time PCR really became recognized around the world is in the field of quantitative PCR. It is considered, in fact, the gold standard when it comes to quantifying nucleic acid targets. Now, over the years, real-time PCR has grown in terms of the number of applications. But from day one, we've been supporting 3 main applications: real-time quantitative PCR, an assay called plus-minus, and SNP genotyping. So we'll begin with real-time quantitative PCR, and we have a rather simple definition here. Now, there are many reasons that you may wish to quantify a nucleic acid target. As you can see on this list, gene expression is number 1. It is the most common usage, but it's not the only usage. I'll take 3 examples from the list. First, gene expression. Typically, researchers would like to know changes in gene expression, say cells, tissue, what have you. And in most cases, the researcher does not need to know the number of molecules of the messenger RNA that's present. They're content to do what's called a relative quantification. So they're comparing sample to sample, expression levels, or group to group. So we would call a non-molecular measurement a relative quantitative experiment. Now, there are many researchers that are interested in quantifying viruses and other pathogens. So in that case, it's more likely that you'll want to know the number of molecules of the viral nucleic acid that are present in the sample, say in a ml of blood or what have you. In that case, we would call that an absolute experiment. There's also a lot of transgenic work that goes on. Researchers may be adding a transgene to, say, mice, and they would want to know how many copies of the transgene are present in the mouse genome. Again, based on copies or molecules, so we would define that as an absolute experiment. Another key application is genotyping single nucleotide polymorphisms, or SNPs. As you may know, there's a growing interest in comparing phenotypes to genotypes. SNPs are involved in metabolic pathways, in disease states, and so forth. And we use a chemistry using a real-time platform to genotype them. So in a traditional biallelic system, you would have 3 genotypes: 2 homozygous and 1 heterozygous. Now, the chemistry that we use for SNP genotyping in real time is called allelic discrimination. And it's very similar to when we use what's called a TaqMan probe to do quantification. But the difference is, instead of one TaqMan probe, which I'll go into more detail later, we use two, one for each allele. So if that allele is present, the one that matches the probe is present in your sample, that probe is cleaved, you get signal. If that allele is not present in that particular sample, the probe then doesn't get cleaved and you don't get signal. So the software then merely has to plot the data as a 2D cluster plot. I seem to have gone backwards for some reason. Strange.
When in doubt, use the key.
Okay. There we go. Okay, the keyboard's working now. Sorry about that. And the third was plus minus. This is a qualitative assay, usually used to detect pathogens, say in food, water, things of that nature. So we're not trying to quantify the pathogen, we just want to know if it's there or not in our sample. Originally, the plus minus assay was intended to be run as a plate read assay. So in other words, you didn't have to collect real-time data, you could read it at the end following regular amplification and a regular cycler. But we do have the option to use real-time data, and that's what we're seeing. That researchers, even though they could run it as an endpoint if they wished, I'm seeing more people prefer to collect the real-time data. The analysis, of course, is very simple. If the target amplifies, it's positive. If it doesn't amplify, it's negative. A fundamental component of real-time PCR is the fluorescent chemistry. Now, there are many of them out there, but certainly by far and away, the two most commonly used are the 5' nuclease assay, commonly referred to as TaqMan, and our company came out with this in 1993, and in 1998 we came out with an alternative called SYBR Green. So let's start with TaqMan. This is a rather simple representation of Taq polymerase. Notice I've represented two protein domains with a red sphere and a green sphere. The green sphere represents the 5' nuclease domain, which has the ability to degrade DNA downstream of DNA synthesis. Another technology that's vital to the 5' nuclease assay is called FRET, or Fluorescent Resonant Energy Transfer. The story behind FRET is there was a German scientist years ago that performed an experiment. Now let's say on the left we have two dyes, a green dye and a red dye. You may be aware that green light has a shorter wavelength compared to red light. So a green dye would be considered a higher energy emitter. So the shorter the wavelength, the higher the energy. Now you might think if you simulate the green dye, you would see a strong green emission, and normally you would. But what Pfister discovered is if you brought the red dye in close proximity to the green dye, now he tries to stimulate the green dye, the emissions are transferred through space to the dye that emits at the lower energy level, the red dye in this case. So in a sense, it's like water flowing downhill, energy likes to find its lowest energy state. So then we could say, on the left, in the FRET configuration, the red dye is absorbing or quenching the signal from the green dye. But, as shown on the right, if we separate the dyes in space, then FRET cannot occur, so now the green dye would emit its normal strong intensity. So if we keep this in mind, and imagine what a 5' nuclease or TaqMan assay looks like. We have two PCR primers, nothing really unusual about them, they're not labeled, but there is a third oligo called the TaqMan probe. On the 5' end is a dye that will report on the amplification of our target, so we call it the reporter. On the 3' end is a quencher molecule. The probe, I'm sure you can tell from the drawing, is gene specific, so it will be designed to bind between the two PCR primers. An important note, the 3' ends of primers, of course, are open and available for extension by Taq polymerase. This is not the case of the TaqMan probe. That 3' end is blocked. It cannot be extended, it cannot function as a primer. And that actually has a lot of importance later on. But before we get to that, if we imagine creating a master mix, so the probe is in the master mix, it's intact, it's soluble. Before the probe gets cleaved, the reporter and the quencher have a natural affinity for each other. They come close together so that if you were to look at the reporter signal pre-PCR, you would find it to be very low. It is being quenched. As I will go into a little more detail later, all of the millions of assays that we have on our website operate under the same conditions. We call them universal cycling conditions. So we use a universal annealing temperature of 60 degrees centigrade. At that temperature allows the primers and the probe to anneal. And you may notice when the probe is annealed to the sequence, it's stretched out in a double-strand configuration. The distance is too large to allow for it to occur. So theoretically, if you had a really fast camera, maybe we do in this room, I don't know. If you had a really fast camera, you might be able to pick up that initial moment when the probe settles down. But the camera would have to be extremely fast because in the next microsecond or what have you, TAC extends from the upstream primer and runs into the 5' end of the probe. Then a key event occurs. If the probe is bound to the correct target sequence, the nuclease activity has the ability to cleave that probe, releasing a fragment containing the reporter dye. So at this point, the reporter is on a separate molecule from the quencher. The affinity between the reporter and the quencher is gone now. So from this point onwards, that reporter molecule that's been cleaved away will stay lit up. So you could say it's a permanent signal production. As you might guess from the drawing, TAC degrades the rest of the probe until we have a second little fragment containing the quencher that also dissociates and floats about. So the two little fragments of the probe will no longer participate in the PCR process, but they'll act as permanent records of amplification. Arguably, specificity is the most important aspect of any assay. And I'm not just talking about real-time, but any assay. When it comes to the 5' nuclease assay, the greatest threat to assay specificity is a homolog. So homolog is defined as a gene which is similar in sequence to that of your target, but it's not the same gene. We don't want any data being derived from the homolog to, in a sense, contaminate or contribute to our results. But the homolog is present in the sample anyway. Homologs, as you may know, are extremely common. They're a natural result of the process of evolution, such as gene duplication and so on. So, how would TaqMan help us with homologs? Let's imagine for a moment that we have a homolog in our sample, which is quite likely actually for most applications. And so it has a fair amount of similarity with the target sequence. The primers would probably bind to some degree to that homolog. And then say there's a base or two mismatch in the primer binding site. One of the problems with primers is that that 3' end is still available. So even though you have a mismatch or two in the primer binding site, TAC can extend. All it takes is a couple of bases extension. That primer binding is now stabilized. It can now, TAC can continue to make product from that primer. This is a problem we have with primers alone. Now what about the probe? Well, the probe, let's say it has a base or two mismatch in the probe binding site. Probe would probably bind to some degree to the homolog sequence. But there's some key differences. First of all, the 3' end of the probe is blocked. So TAC cannot extend the probe. It cannot stabilize that mismatch binding. So when TAC strikes the probe is displaced from the homolog. So this mismatch creates what's called a delta G change, preventing the probe from being cleaved. So instead, it's displaced from the homolog, recoils back to its original quenched configuration, so that at the end of the PCR cycle, which is where we collect all of our data, you're not going to detect signal from homolog, you only detect signal from the target. A powerful benefit of the TaqMan probe technology. Now, many of you are probably familiar with PCR, and you're probably familiar with the concept that PCR often produces things called non-specific products. Perhaps the most famous of which is the so-called primer-dimer. That is, the primers are binding not to homologs, but they're binding to just random sequences, maybe even to themselves. And they will produce various random type products. Now, of course, since these random products have no relationship with the target sequence, there is no probe binding site whatsoever. So a TaqMan probe could not bind to a primer dimer, much less get cleaved. So automatically, non-specific products are excluded from our data using the TaqMan probe technology. Now, it may be interesting to note that we offer two different types of TaqMan probes with different specificities. Originally, TaqMan probes had a TAMRA dye on the 3' end. Now, we still make them, but the fact that the quencher is TAMRA is not really the key issue here. The fact is these probes were longer than primers, which meant that a single base mismatch in the probe binding site had very little impact on whether the probe would get cleaved or not. The probe would probably still get cleaved, as a matter of fact. Maybe even 2 mismatches, the probe would probably still get cleaved. Now, 3 mismatches, probably not. If you go to our website, you'll see that the vast majority of our TaqMan assays are designed to eukaryotic targets, like human gene expression, SNPs, microRNAs, and so forth. Now, eukaryotes have a low mutation rate. So there's a lot of sequence homology, a lot of homologs. Great example, gene expression. There are a lot of homologs there. Very few base mismatches between the homolog and the target in many cases. MicroRNA is another great example. MicroRNA genes are highly conserved evolutionarily. Many genes only differ by 1 base. So our researchers wrote this older style of probe gave an insufficient level of specificity for the targets that we want to design assays for. So this is where they came up with the TaqMan MGB probe. The MGB probe has a non-fluorescent quencher on the 3' end. Again, the fact that it's non-fluorescent doesn't matter that much. What's more important, there's a second molecule attached to the 3' end called the minor groove binder, or MGB. So if you can imagine the TaqMan MGB probe binding to the target sequence, a short double-strand region is formed naturally, and that forms a small minor groove, which allows the minor groove binder to bind, strengthening the binding of the probe to the target sequence. The result of this is we can design MGB probes that are shorter than primers, which means now that 1 base mismatch in a short probe has a much greater impact on whether the probe gets cleaved or not. So this is why you will find on our website the vast majority of our assays, which are eukaryotic targets, use MGB probes to get the specificity that we need. But perhaps some of you may be working with other targets, prokaryotes, for example, which have a higher mutation rate, maybe viruses with a high mutation rate. Maybe you want to allow a base or 2 mismatch in the probe binding site and still get signal. Great. In that case, a TAMRA probe would be a good selection. But either way, whether you have a TAMRA probe or an MGB probe, the same principle applies as far as signal accumulation. So what I'm showing you is an amplification plot. So we have what's called the delta Rn. Think of it as a fluorescent signal on the vertical axis, PCR cycle number on the horizontal axis. Now initially, you know, we have a flat area we define as the baseline. So the baseline, whoa, I didn't do that. The baseline is defined as the early cycles in PCR before amplification becomes visible. So you can guess from this that amplification must have been happening in the baseline cycles. But in these early cycles, we have not accumulated sufficient cleaved probe to see the signal above the noise. But by the time we get to this point, say around cycle 26 or so, we have accumulated sufficient cleaved probe. We are now seeing strong signal emerging from the baseline. So there's a little rule that's going to set the stage for quantification. That rule states that the greater the number of target molecules I start with before PCR begins, it will take me fewer cycles before I cleave enough probe to see signal. And conversely, the smaller number of target molecules I started with, it'll take me more cycles before I cleave enough probe to see signal. So we could call this an inverse relationship, and that forms the basis of quantification in real time. So before I get to that, I wanted to mention another chemistry that we came out with in 1998 called SYBR Green. So SYBR Green is a minor groove binder dye. It just binds to any double-strand DNA. So in a sense, it's like Ophidium in that it just binds to DNA. There's no specificity involved there. So a SYBR assay would consist of 2 primers. There would not be a probe. A similar concept with accumulation of signal occurs with SYBR. We need to have a certain number of cycles go by before we have accumulated sufficient PCR product that we can see the signal about the noise. However, it's important to note that there is controversy attached to SYBR Green. And as you might guess, it surrounds the issue of specificity because unlike TaqMan, there is no probe to protect you from specificity problems. Anything that's double-stranded DNA, CYBR will bind to it and will contribute to your data. And in fact, there have been researchers who have done comparisons between TaqMan and SYBR results with the same target, the same samples. And the concordance in those results has been very, very disappointing. And the evidence seems to point towards specificity as being the culprit. More controversy, how do you determine that your CYBR assay, or I should say this way, how do you measure the specificity of your CYBR assay? I don't think there is a good solution at this point. I mean, there are things that people do, but none of them, I would argue, are absolute. Some people sequence, but there are issues there. I'm not going to go into a lot of times all the different reasons for that. Some people run gels. You know, helpful thing to do. Sequencing is helpful. Gels are helpful, but again, not absolutely conclusive. For example, if you were amplifying a homolog, there's a good chance it might have a similar molecular weight as that of the target. It may co-migrate on your typical lithium bromide stain agarose gel. It's definitely has been known to happen. But certainly the most common thing that researchers do is what's called a melt analysis. So the concept with a melt analysis, and we started talking about this back in 1998, is that if you had a primer dimer, let's say, then by chance it will likely have a different melting temperature compared to the target. I think that's a fair statement. And there are exceptions. Primer dimer has a similar melting temperature as the target, but that's the exception. The way it works is, by the way, the melting, following PCR, the instrument will start at typically 60 degrees centigrade, where the amplicon should be double-stranded. And so if you have positive samples that amplify, you should see a strong CYBR signal, which is shown here. As the temperature increases, oh, you can't see my pen, can you? As the temperature increases, I'm using the left panel. Use the mouse. Okay, great.
Mouse down, we'll see it.
There we go. Okay, great, thanks. We see a drop in CYBR signal. Now, this initial drop has nothing to do with the amplicon. This is just the decrease in fluorescent signal that you get with increasing temperature. And where the drop accelerates, that is the amplicon or amplicons that are being melted. And eventually it bottoms out at very high temperatures. Now, the melting temperature of whatever this is would be defined as the inflection point on this graph. That may be a little difficult to see, but good news, the software takes the negative first derivative, converting the inflection point into a peak. So then, in theory, you should see one peak, and you're hoping that that is the target. But things are typically much more complicated than that when it comes to CYBR. You often get various anomalies. And if you see a dissociation anomaly, such as an asymmetrical peak, is shown here on the left. Oh, I'm sorry, it's shown in the middle. Darn it.
Yeah, I touched that.
We have another peak over here. It's a different peak than the main peak. We have an asymmetrical peak, clearly the fusion of two peaks to each other. And then this is pretty obvious, we got three peaks. 2 peaks is bad, 3 peaks is really bad, I guess. But also, I should point out that even if we see what might be termed a pristine peak, what appears to be a single, narrow, has to be narrow, symmetrical peak, we cannot assume that is the target, nor can we assume that that is one product. Why? CYBR melt is low resolution, not high resolution. It's never designed for high resolution. So the danger is it may look like one thing, but in reality, it could be multiple things. So in the initial data that we were showing in 1998, we would use things like plasmid. It has very low genetic complexity, and then you have primer dimer. If you saw a peak with a plasmid, pretty good chance that's the target, I would think. But the vast majority of applications involve high complexity samples, like total RNA, total cDNA, total genomic DNA. So the probability of homologs there, very, very high. So again, just because you have what appears to be a single narrow symmetrical peak, you cannot assume that is one product, nor it is absolutely the target. Now I'd like to switch gears from chemistry to instrumentation. Over the years, we've had many different models of real-time machines. Here's some of our current models. Don't want to go into a lot of detail, but I'll choose two from this list to go a little more depth. The 7900, which is highly capable. We use a laser, emits light at 48 nanometers, which stimulates the entire either 96 or 384 well, depending which block that you have in there, so that the entire scan takes about 8 seconds per scan. The laser light stimulates the dyes. Those dye emissions are then passed through a device called a spectrograph. Perhaps you're familiar with how white light passes through a prism, and then it stretches, in a sense, the EM spectrum into the different colors. If you recall from high school, Roy G. Biv, remember that one? So think of a spectrograph in that way. It's stretching the EM spectrum, and so we're detecting the entire spectrum between 500 and 660 nanometers on a CCD. In contrast, the 7500 uses a halogen-based excitation. So it's broader excitation wavelengths, and those excitation energies are passed through 5 filters in a filter set, filter wheel. The emissions are then passed through 5 emission filters in a filter wheel and detected again on a CCD. But all of our instruments have at least one thing in common, and that is an algorithm called multicomponenting. So what we do is recognize the fact that dyes are not going to emit through a single filter. If you look at dye spectras, they're more of a mountain shape. And so multicomponent takes that into account and sums up all the contributions across the filters for specific dyes. Now you don't have to worry about the math. It's very math intensive, but don't worry about that. It's just built into the software, and you get to take advantage of it. So one of the things I like to reassure my customers is that a lot of people are concerned. Well, what if I didn't label the dyes correctly? Is my run ruined? The answer is no. You can always go back to the data file, put in the right dyes, and reanalyze. This is one of the benefits of using a mathematical algorithm to calculate pure dyes instead of having to pick and choose which filters are going to collect dye signals. So this is a nice benefit. Of course, we don't tell people that you have to buy more filters. But this is number 3 is my big one. People send me their data frequently, and I troubleshoot, and I use this multi-component data to troubleshoot all the time. I just used it, in fact, a little while ago. Here's a very simple example on the left. The dye designation is VIC for the reporter, if you look in the lower left corner, and ROX is our passive reference dye. So we use a red dye called ROX in all of our real-time master masses. It serves many useful functions, including, as you'll see here, troubleshooting. Now initially, the red line, the ROX line is flat, as it should be. So far, so good. But then you'll notice as the VIC signal grows, the ROX signal decreases in a kind of mirror image. There's no chemical explanation for that. There's only one possible explanation. It's got to be a dye designation error. So VIC must not be the correct dye. So changing it around, shows FAM, which is shown here on the right, reanalyze, and now ROX is perfectly flat as it should be, and I know I've solved the math error. Therefore, FAM was the correct dye, not VIC. I mentioned a moment ago that all of our assays, and that includes the ones on our website and assays that perhaps you've developed yourself, if you used some of our systems. There are 2 that we have, one called Primer Express, the other one is called the Custom TaqMan Assay Design Tool. Now, all those different options, the assays will operate under universal cycling conditions. Also, universal magnesium, universal primer and probe concentration. So we've been able to eliminate optimization. Our universal cycling consists of a 2-step PCR, which can be used on gels. Of course, we do it all the time for real time. So we're using a 60-degree step as both annealing and extension. Realize that real-time amplicons tend to be rather short. So the need to do a 72-degree extension is not there. In fact, it may be possible that many gel-based assays, 72 degrees is unnecessary as well. Now, initially, we had a certain ramp mode, meaning ramp is the change in the temperature in the block. And the majority of the ramping that was done is what is called standard mode. But several years ago, our researchers developed a new way to ramp called fast mode. So the cycling conditions is still a two-step between standard mode and fast mode, but the hold times are a bit shorter and the ramp rate is considerably faster in fast mode. So if you have a fast mode capable real-time instrument, that would automatically mean you'd be using fast plastics, fast plates, for example, and you would use fast master mix. So in standard mode, 40 cycles would take about 1 hour and 3/4, whereas fast mode, 40 cycles would take about 35 minutes. A lot of people are enjoying that. Data quicker. Of course, real-time assays, though, are the same. We don't have fast and standard assays, but the assays are the same. So I'd like to now switch gears a little bit and talk about assays a bit. And of course, we talked about the two major types of assays, the 5' nuclease and SYBR Green. When doing an experiment, it's important, I think, for most applications, those dealing with biological samples, that we have, in addition to the target gene, the gene that we wish to quantify, that we also use a gene to normalize our data, normalize based on sample mass. So I often use the term a normalizer assay or normalizer gene. As a good example, in gene expression, we'll use genes, perhaps you've heard of them, 18S, GAPDH, beta-actin. Some people use the term housekeeping gene to normalize based on sample mass, RNA mass in this case, and other useful functions. So years ago, our scientists created a system and began by establishing universal cycling conditions. And over a period of years, they did massive systematic wet chemistry testing to identify the design parameters and chemistry formulations that would allow all the real-time assays to run under universal conditions and run robustly. So all assays, whether designed by us or perhaps by you using our software like Primary Express, will run under these universal conditions and you'll get those benefits. In general, we divide the choices into two categories for assay design. TaqMan assays are assays that we've done all the design, the testing, the bioinformatics. They're on the website, ready to go. Custom assays are any assays other than that. So traditionally, for many years, researchers use software that we provided called Primary Express to design the assay. So that way, you would get your 100% efficiency, universal cycling conditions, universal magnesium, and so forth. But Primary Express didn't do any bioinformatics. More recently, we have now a web-based tool called the Custom TaqMan Assay Design Tool. And this tool now offers a new feature that we've never had before, the ability to design a custom assay where if the organism, the species that you're working with, is one of the ones that are covered under the Custom TaqMan Assay Design Tool, we'll actually do the bioinformatics for you. No additional charge. And so that means we're getting the same specificity for a custom assay as we do for a TaqMan assay. That's powerful. We've never offered that before. I think it's a great tool. But to focus on the TaqMan assays for a few moments, most of the TaqMan assays that I encounter are gene expression, but realize we have many others. MicroRNAs, SNPs, copy number variation, and the list goes on. So for gene expression, we currently cover 19 species plus a handful of viruses. And we display the bioinformatics on our website. For example, what you're seeing here is called the GeneAssist Alignment Map, which I think is a great way to look at the transcript structure of the gene that you're studying. I'm not going to go through all the different ways that the TaqMan assays were designed, developed, tested, and all that. It's a lot of work there. But the good news is, while it's probably impractical for an individual researcher to put that level of effort into their assay, But you don't have to, because when our researchers put that level of effort into the assays, the cost of that gets amortized out around the world as the assay is purchased by people around the world over time. So you don't have to pay for that. You don't have to reinvent the wheel. So this is really good news. But all of that complexity in the work, in a sense, you could say are behind the scenes. But what you get is a premix, 2 primers and a probe, typically a 20x when it comes to gene expression. In cases where you want to quantify, shall we say, a small number of genes, and according to my math, that would be less than 12. So the breaking point is right around 12 assays. So less than 12 individual assays, great choice, that's the way to go. But when you wish to quantify more than 12, it becomes more economical and certainly way easier to do what are called TaqMan arrays, or TaqMan array plates, where the assays are already dried down into the plate. So all you have to do is make one master mix. I mean, imagine if you wanted to quantify 96 different genes, if you did it with individual assays, it would be really expensive and it would be a lot of labor. You have to make 96 master mixes. This way you only make one master mix. So as I mentioned, for custom assays, whereas for years we've been talking about Primer Express, but now because the custom assay design tool allows you a whole new set of options that you never had before. The option to generate assays, they're in that 20x premix, so it's really easy to use, minimizes error potential, minimizes time, and so forth. Also, if it's one of the species that we cover, we'll do the bioinformatics for you. And we now have choices. Choosing between FAM, Vic Universal Primer Concentration, or Vic Primer Limited, for those who are interested in multiplexing. And things keep changing, so over the years, we've seen large growth in the number of real-time master mixes that are being offered. And same is true of us. So we have a number of TaqMan master mixes as well as a number of SYBR Green master mixes. They typically come as a 2x concentrate. So these master mixes will contain your TAC, the buffer, DNTPs, etc., but they won't contain the primers or the probes. And for those who are doing work with RNA such as gene expression, of course we need to think about reverse transcription. We can divide reverse transcription chemistries into 2 categories, called the 1-step and the 2-step. In the 1-step, we add RNA into a single master mix. Reverse transcription occurs first, and without you having to do anything, it immediately moves into PCR. 2-step is where you make cDNA first, usually with random primers, not always, but typical with random primers. Then you have to physically transfer the cDNA into the PCR master mix. Now I'd like to move into the area of analysis. So let's start with the foundation, which is the 3 phases of PCR. For those who are new, we have 3 phases of PCR that if you let it, PCR will progress through. The first phase, you can term it geometric, logarithmic, or exponential. Doesn't matter, each term is synonymous. It's in this first phase that the amplification efficiency is consistent. And this is the phase that we focus on for real-time quantification. But eventually, PCR will transition to the second phase, called linear phase. So in linear phase, the efficiency keeps declining cycle to cycle. So that means a reagent in the PCR chemistry has become limiting. If you plot the linear phase, it will appear as a curve. If you let PCR go long enough, we may eventually reach plateau phase. This is where the limiting reagent has become exhausted. PCR comes to an end, the plateau phase will look approximately like a horizontal line. I like to argue this slide is in fact the reason for the success of real-time PCR, and arguably why I'm here today. And that is this principle, this characteristic, if you will, of the geometric phase. And that is, no matter how many molecules of the target are present in your sample, all the geometric efficiencies within that assay are the same. Doesn't matter if you start with 10 copies, 1,000,000 copies. the geometric efficiencies are the same. Now that would imply, as shown here on the left, that if I were to plot the geometric data in a semi-log plot, the line should be parallel. And in fact, that's exactly what we see, parallel lines. So the next step in the process is we draw a horizontal line through the geometric data, which we call the threshold. Next, where each of the geometric applications cross the threshold, this will correspond to a PCR cycle number that we call the threshold cycle, or CT for short. Now realize, while I've drawn the CTs as whole numbers, in reality, they're more likely to have decimals. I'm just trying to keep it simple. Now if you look at this graph, you may say, you know, I see a pattern here. Notice how the lines are forming a pattern. And if you see a pattern in a graph, that implies a mathematical relationship. In fact, there is a mathematical relationship between the initial quantity of the target and the CT value, which is shown on the right. So the initial target quantity is proportional to the geometric efficiency, designated as lowercase e, taken to the minus CT power. So I don't want to, of course, bore you with the math here, but in the grand scheme, what's happening is the technology is going to deliver CTs. If you designate the efficiency, so we got the E and we have the CT, we can then deduce mathematically the initial target quantity. And it's because of this fundamental relationship between those two. I very strongly recommend to all my customers that when starting a new assay, that you identify the dynamic range of that assay. So dynamic range can be defined as the minimum and maximum sample amount that can be safely run in your assay system. So if you run samples outside of dynamic range, while you'll undoubtedly get CTs, although there may be exceptions, usually you do get some kind of CTs, but the CTs will no longer be accurate and may not be precise any longer. So we need to stay within dynamic range. And there's many factors involved in dynamic range, such as the quality of the RNA, if there's inhibitors present, reverse transcription issues, that means that you have to empirically determine dynamic range. You can't just assume it. Once you've completed dynamic range testing, so now you know what the dynamic range of your assay is, this will enable you to take steps to ensure that all your samples, as best as you can, will be measured within the dynamic range of your assay. So at that point, you can stop running standard curves, which is what we do to test dynamic range, stop running them, and instead switch over to data production using the delta-delta-CT method. So the delta-CT method is a method our scientists invented years ago, and the equation it's based on is shown here. Now, I hope that you see that there's a similarity between this equation and the fundamental equation I showed you a few slides earlier. And that's exactly what it is. This delta-CT equation is based on that fundamental relationship between the CT and the initial target quantity, as I showed earlier. So this is not a line equation. There are no standard curves on the plate. So that means you've reduced your labor, you've reduced the cost per data point, and, very nicely, you've removed a source of variability in your data. Now, delta-CT does require assays to have 100% geometric efficiency, but guess what? All assays designed and run in accordance with our recommendations do have that. They all are 100%. And that completes the seminar. So thank you, and I'll be happy to take any questions. Yes?
So you suggested that the delta-CT method, once you've established a standard curve, you can have that as in memory and don't have to repeat it every time you perform an experiment?
Okay, great. Great question. If you'll allow me, I'll kind of rephrase your question a little bit as I understand it. That you're saying, is delta-CT dependent on running an initial set of standard curves, recording that data, and then using it in further analysis?
For the same set of, let's assume, for the same amplicon.
Right, for the same assay curve, yeah. Answer is no, which is good news. So if you'll notice, again, delta CT equation is not a line equation, so there's no standard curves that are involved in the math. What I was recommending in this slide was to use standard curves not for purposes of calculating geometric efficiencies, because we know there's a lot of problems with that. Because when you create standard curves, of course, you have to pipette. And I'll just use myself as an example. I think I'm pretty good with a pipetter, but I'm not going to say I'm perfect. Even a robot's not perfect. Mistakes are going to happen. It's not your fault, it's not anybody's fault, that's the way it is. Mistakes in the standard curve will throw off the slope. Well, if you throw off the slope, you're throwing off the implied efficiency of the assay. So it means that the slope is no longer reflecting the true efficiency. So this is what I was saying. Standard curves, if you use them for the purposes of data production, converting CTs into quantity, you're actually introducing a whole level of variability, of error into the results. Fortunately, with delta CT, we can eliminate that source of error by not using standard curves in those equations. In this equation, there are no standard curves involved in this. So I think this is good news. That while, again, I do encourage people at the beginning of a new assay system, which includes everything, extracting, RT, PCR, that's the system. Test uninfringed. If you're doing gene expression, for example, these would be RNA standard curves, not cDNA, RNA standard curves. And look for things like inhibitors. Look for the tailing effect. See if there's problems there. Look at those slopes. Are they really horrible slopes? Are they really shallow? Or they look decent? They're somewhere -3.3 in that ballpark. You look for things like that. Make sure everything's working smoothly. And then forget about the standard curves and move into data production with delta CT. But does that make it clear? Or, you seem to have a little bit of a frown there.
I'm just trying to figure out. Eventually, you would want to assign a absolute number as opposed to this is probably going to come up as a relative number.
Oh, okay, great. Another question. So is delta CT, I'm going to, again, kind of rephrase your question. Is delta CT only good for relative quantification? Can it also be used for absolute? More good news. The answer is yes. You can use it for both relative and absolute. Give you an example. We have free software on our website called Copy Caller. I don't know if you're familiar with copy number variation. In the human genome, we have segments of our DNA that may be either missing or they may have amplified a little bit. 3 copies instead of 2 copies, for example. In fact, I do a mini course for junior high students where that's exactly what we do. In any case, we use the delta CT method in the Copy Caller software. And what we do is we select an absolute calibrator. So that's a sample where we know what the copy number is. So it'd be 1 copy per diploid, 2 copies, whatever that is. We know what that is. And that creates AQ, absolute quantities, for all the samples. So yes, delta CT may be used for relative or absolute. It just depends on the calibrator that you select. Obviously, relative calibrator for relative quantification, absolute calibrator for absolute calibration.
So in a real-time setting, if you were to obtain absolute quantities of a bottle genome,
Great example. Okay.
how would you go about doing that?
Great. Allow me to ask you a question so I can tailor the assay to your application. What type of sample would you be extracting the virus from?
Let's say infected cells.
Great. Okay.
It would
Human?
be, let's say, simply not an RNA virus, but a DNA virus.
DNA virus. Okay, great. Like adeno, perhaps?
Yeah.
Beautiful. In fact, I've been working with some of those assays recently. So would you like then to have the results as copies of adenovirus per copy of human DNA? Would that be reasonable? Or human cell number?
We could do that, or even total absolute number. For instance, in virology, do a lot of particle to infectious units, particle to PFU.
Ah, okay.
So basically, you want to do the genome per infectious particle. So you want to know the absolute number of genomes that ultimately do create an infectious.
Oh, okay. No, that's a cool experiment. I like those.
Particle to PFU is called.
Yeah.
But I mean, I think both would be interesting. An absolute number, if I was to determine what are the absolute number of genome equivalents in that sample, I would also translate that to the number of genomes per cell, because I know the number of cells. So I guess you could probably arrive at the two. That's what I'm trying to achieve.
Yeah, both are possible.
Yeah.
Both are possible. So the assays, the adeno assays I've been dealing with recently, the researcher wanted to know how many copies of the adenovirus were present. You can either say on a per cell basis or per genome copy number, however you want.
Yeah.
That's easy enough. 2 copies per cell, all right? There you go. So what we did is we used the RNase P assay as a measure of the number of copies of human DNA. And we used, of course, an adeno assay to measure the adeno. And the researcher had, I believe it was a cloned version of the adeno target. And so, of course, you pre-quantify that with OD260, do the stoichiometry on that to turn it into molecules. And then he created an absolute calibrator. So he could take a known number of molecules of the adenovirus, mix it to control human DNA. Actually, we sell that. Pretty cheap, but it's good quality. So we know the exact ratio. In fact, I think it's really cool if you can make the ratio 1-to-1. Because if the ratio is 1-to-1 for your absolute calibrator, if you look on our Delta CT software, it'll use the term RQ, relative quantity. So the calibrator is 1. Well, if the absolute calibrator is 1-to-1, you multiply 1 times 1-to-1, you've got AQ. RQ times your multiplier gives you AQ. RQ is AQ. Makes it really nice. So I'm not saying it has to be 1. It's whatever you want. That's one approach. Now, when it comes to the PFU, clearly, you're going to have to do some work with saying, okay, I can do a PFU assay, which is kind of separate from the real-time. But using the same stock of virus, quantify the stock and say, okay, according to my real-time data, I've got this many molecules per microliter. And then I'll say, okay, this many molecules of adenovirus genome produces this many PFUs.
So you still think the relative CT would be better than having an absolute curve, actually measuring the amount of micrograms of DNA and doing a standard curve?
Well, what I'm saying is dynamic range testing is a separate function from producing data. So what I'm proposing is using standard curves to produce data is not ideal. There's a list of reasons why. Errors, they cost money, they occupy space on the plate, and etc. So, for example, if I was doing dynamic range testing in an adeno experiment, say extracting adeno from human cells, I would take the human cellular DNA extract, do a serial dilution, and I would look for things like inhibitors. Now, obviously, with the human DNA, we don't have to worry about reverse transcription. That's irrelevant. But we do have to worry about inhibitors. That's an ever-present concern. But then once I've identified, oh, look, there's no inhibitors. Yay. So now I can go straight into data production using Delta CT. Happened again. What? Oh, I'm going the wrong way, sorry. The data production using Delta CT, stop worrying about the standard curves. Now, of course, if there's something happens that you think may have changed the scenario, let's say you've gone from this extraction method to a totally different extraction method. Maybe dynamic range has changed now. But I think as long as you're using a consistent methodology, the same kit, you've got things QC'd, so like an ancient kit may not work as well as a fresh kit, things like that. Then dynamic range should remain stable. Everything's being carefully monitored. Look at your RNase P, make sure you're getting a reasonable CT value there. That's another way to monitor your dynamic range. Then there shouldn't be a need to keep repeating dynamic range, so you can stop running those standard curves. Of course, unless you suspect there's a problem, then go run a standard curve and start to investigate that problem. So here's the goal is to reduce cost, reduce labor, maximize accuracy and precision.
Of course, you would argue that if you had purified adeno DNA, that wouldn't be really equivalent. So you couldn't do a standard curve on purified DNA.
That's a good point. I'm glad you brought that up. I was thinking about talking about it, but you brought it up first, thanks. So the idea would be if you test dynamic range, of course you have to use a representative sample, something that is just like all the other samples in your experiment. If you take purified, say, plasmid, like you were suggesting, well, plasmid is going to be really clean, I think, especially if you dilute it 1,000,000 times to get it down to a reasonable copy number. So you'll never see, probably, I would think, an inhibitor in plasmid. But if you extract total DNA from cells and tissue, well, sure. I mean, those could have inhibitor. So, yeah, for purposes of dynamic range testing, pure template, what would mean? Pure template, like cRNA, plasmid, artificial DNA, things like that, those are not going to be a valid dynamic range tests.
I
Yes.
I have one more question about
Only one more?
Yes, one more. When we use the threshold, and compare that to using, say, a standard curve with known amounts of plasmid with serum dilutions or known amount of applicon with serum dilutions to determine the absolute number there.
Okay.
There are methods where some people use the absolute number to determine copy number, whereas there are some where there is a comparison used based on cutoff, where it crosses the threshold frequency or threshold, where the curve crosses the threshold cycle.
You mean here?
Yes.
Okay.
So when you want to compare samples using just the threshold,
You mean the CTs?
yes. How is it possible? Is it possible to assign a number, or you just, in this case, we'd probably say it crosses at, say, cycle 26 and, say, 28 or something to that effect.
Okay.
Now, is it possible to assign, aside from the cycle number, is it possible to assign a number to compare those two samples? You know what I'm saying? One of those samples that crosses early has possibly higher starting material, whereas the one that crosses later has less. So point I'm trying to make is, if you have two samples with different amounts, but you don't have your amplicon clone to produce the standard curve, will there be any way of depicting the difference without using the standard curve, just using the where it crosses the threshold?
Right. You're perfectly describing the delta CT method. It's perfect, thanks. And I didn't go into details how it works, but now that you've brought this up, this is a good opportunity. So you'll notice there are two deltas in front of the CT. The first delta, and by the way, delta implies subtraction. That's what that means. So we're subtracting pairs of CT. So the way it works is we take the target CT, say for sample number 1, and subtract from that the normalizer CT for sample number 1. And that creates the delta CT. So this would normalize based on differences in sample mass, sample to sample. So if one sample had, say, twice as much RNA as compared to the other sample, then that CT for the normalizer will be 1 smaller. So that gets determined in the math. Now, the second delta is we're going to choose a sample to act as our calibrator. So that sample will be assigned a RQ, or relative quantity, of 1. So that at that point, that's an arbitrary 1. All other sample RQs will be relative to that 1. So that second function in a relative experiment is very minor. It's just helping the data to be a little easier to understand. Now, of course, in the example you gave with absolute and viruses, things like that, the calibrator serves a much more powerful function, and that is to provide absolute calibration. So in that case, you definitely need the calibrator, and it's an absolute one in that case. Whereas in the case of relative quantification, the calibrator arguably is optional, even. But you're exactly right. That's the first step in the delta CT equation. We must normalize for differences in sample mass, sample to sample. And that's what that first delta CT is. Great, thanks.
So what would it be represented as? Let's say they're crossing at cycle 28 and 32 or something.
Mm-hmm.
So how would that difference be represented?
Okay, so now realize in the delta CT method, there's a series of mathematical steps. So we have the delta CT, that's step 1, then the delta delta, that's step 2, and then conversion of delta CT into quantity, that's step 3. So there's 3 mathematical, if you don't count the averaging for your replicates, then there's 3 key steps in the delta CT method. So the final result is, in this example, is RQ, relative quantity. So if you're just picking any random sample as your calibrator, so assign a value of 1, well, we don't know what that 1 means. We don't know if it's 1,000,000 copies, 1,000 copies, but then again, we don't care either. If it's a relative experiment, we just don't care.
It's across the board, common for all samples.
So you standardize one and you refer comparing that to that particular one. So
Exactly. Exactly.
All of that.
That's right. So we can say, well, this is, our calibrator is 1, RQ of 1, the sample is an RQ of 2, then obviously the target was expressed twice as much in this sample compared to the
That value is calculated for every time, is automatically calibrated or calculated by the system?
Well, that's a great question.
Or is it assigned?
Yeah, how is it assigned? If you don't do anything at all, the software will choose the calibrator alphabetically. After all, it's a relative calibrator, it's arbitrary. Now there is an exception. If the calibrator fails to amplify, then that's not going to work mathematically. So, you know, if it did pick 1 that didn't amplify, then you need to change that to a sample that did amplify. But it's so simple in the software. You just pick and choose any sample you want to act as a calibrator, reanalyze, that's it. You're all set to go. So you have full flexibility in the software to choose a different calibrator, you know, if you see fit to do that. But realize, if you change a calibrator, for relative again, it's not going to change the quantitative relationships among your samples 1 bit. All it's doing is it's changing the numbering system. So don't be afraid. You want to change the calibrator? Great. Obviously, absolute calibrator, that's a different story.
So what do you normally use for absolute calibration? What is the internal standards?
Okay, now of course the absolute calibrator will depend on the type of result that you want. For example, if you wanted to do absolute gene expression, if we want to know the molecular concentration of the messenger RNA in, let's say, on a nanogram basis of total RNA. So in that case, what I would do is I would make in vitro transcribed RNA for my target. For example, Ambion has a kit called Megascript that does that sort of thing. It's pretty simple to use. So you're determining, you know how many molecules of the in vitro transcribed RNA you have. Then you add a known number of molecules into a known RNA mass of control RNA. Now when you do this, just make sure that the number of molecules of the in vitro transcript far exceed, I'd say 100-fold or better, any endogenous target that might be present in the control RNA. And that's it, you've got it. That's your absolute calibrator. And it has to be RNA in that case because we have to take into account reverse transcription efficiency.
So for DNA sample, going back to the
Adenome example.
what would you do? Spike DNA onto the actual index?
Yeah, so again, it depends how you want to normalize your data. Like in the example I gave where the researcher wanted to normalize the data based on copies of human DNA, then you take the control human DNA, you mix in a known number of copies of the pure adeno clone, there you go, you got it. On the other hand, if you're looking for, let's say, adeno in a liquid, maybe blood or something, in that case we would use an exogenous normalizer. So things like RNase-P, that's an endogenous typically. So exogenous would be like, we have an IPC, exogenous internal positive control. You could spike it in so many either mils or not mils, microliters per mil of blood prior to extraction. So that way, when you extract the DNA from the blood, the efficiency of extraction should be the same for the target and the normalizer, so it corrects out in the math.
Well, there's going to be a pipetting error there because you have to still put.
Well, you're right. But that's why we do replicates. So, but if you do replicates, that'll help minimize that. We do the best we can, right?
Any questions at LSUS?
universals?
It wasn't on right at the beginning, it's on now.
Okay. You mentioned that your assays are
It's
universals.
too loud.
Oh, it's too loud?
I can barely hear her.
Yeah,
She's going to turn it up.
Can you hear me now?
it's,
Can you
not-
turn
it's-
it up a little bit?
it's not picking up at that mic, it's picking up at a different mic. I can tell. I do this.
Okay, you can hear me now, right?
Yes, now it's on.
Thank you.
You mentioned that your assays are universal, so can I use those assays with any thermocycler that can perform real-time PCR? Like, for example, the Realplex, the Eppendorf master cycler, could that be?
I mean, it should. The vast majority, well, first of all, the question is the unit that you're talking about, can it handle the dyes? So, for example, for gene expression, we use FAM typically. But we do, like for SIP assays, we use FAM and VIC. So then you'd have to ask the question, can that unit handle a FAM-VIC in the same tube?
Okay.
So that's one thing you would want to look at. Temperature-wise, you would think, if you said 60 degrees here, 60 degrees there, I mean, in theory, it should be the same temperature. Now, as far as ramp rate goes, you know, I'm obviously a little more hesitant on that one. You'd have to sort of dig into that to see what kind of ramp rate are they offering. Is it similar to our standard ramp rate, or is it more similar to the fast? And just kind of investigate that.
Okay.
If it's standard, then you can use a standard master mix. Fast, then you'd want to use fast master mix. So I think those would be the two main things: optical issues and thermal issues, meaning ramp rate.
I think most of those suppliers make those machines to be compatible with ABI chemistries.
I think that's a fair statement. Yeah, I think it's a fair statement. And I have seen exceptions. For example, there's an instrument that uses glass capillaries. I've seen some issues there. But for most of them use plastic plates, and I think that's good. I mean, a polypropylene plate is usually a standard thing people use in PCR.
Any other questions, Tara?
No, that's all. Thank you.
How about from Louisiana Tech? Do you guys have any questions?
June? No?
I don't. Thanks.
No, no question.
How about that?
Okay.
So, I
All
guess-
right. How about here? Any more questions here? No?
I'm sure John will be around to resolve some problems and have some specific things.
I do have a flight to catch, but
Yeah. Okay. Thank you, John, very
Thank you so
much.
much.
Appreciate it.
my pleasure. My pleasure. Thanks.